right place if you. So if you are here you're here for Dr. Pen's talk, Diet, Microbiota, and the Cancer. So I would like to first introduce about, say something about this Zoom talk. This one is co-hosted by CBA, Chinese Bioscience Association, and also New Hope Chinese Cancer Care Foundation. I would like to invite Dr. Pen as a president to give a few words about New Hope Chinese Association. Dr. Pen, are you there?
Thank you. Yeah, I'm here. Thank you, Su-Ming. And thank you everybody for taking your precious time on a Saturday morning to join us. I'm Minggui Pan, and I'm a volunteer and president of New Hope Chinese Cancer Care Foundation. We are a nonprofit organization serving Chinese cancer patients and their families in the Bay Area. Our executive director, Tina Tai, is also here with us today on the line. Su-Ming.
Thank you. Thank you for a very nice, brief introduction. I will give you more time to talk about New Hope later in a big time. So let's just go on. So here, CBA CBA welcomes everybody who already followed our talks all the time. I welcome all the old friends and also new friends. So I am Shu-Ming Liu, the president of 2021. So I give just a very brief introduction about the Chinese Biosciences Association, CBA. CBA was founded in 1997. As I mentioned for the last few talks, it was founded by a few Stanford postdoc students in a garage, but now it's very big. It was founded in San Francisco. That's how you can see it from CBA SF. This is where SF comes from, .org. If you want to know more information, I would suggest you go to the website and check out. We have very exciting programs. So I will talk about it in a few minutes. So this is a bridge that everybody knows it's in San Francisco. We were founded in San Francisco, but that won't limit us to reach out. So CBA is basically acting as a bridge to connect the people together, to change their thinking, their ideas, their results, to discuss future. A lot of things happened in CBA. So our members, I just listed some of them: CEOs, business executives, professors, scientists, postdocs, PhD students, etc. Even if you're not belongs to any group of these, you are welcome too. I know some of us in the audience probably never belongs to any group of them, but they followed followed our talks all the time. I feel very happy. This is a map I borrowed from the airline. Doesn't mean we already reached out so many places, but we're working on it. Not only the places as a distance, but thanks for to the internet and the Zoom conference, all these. So we were able to reach out. Right now we have audience in the audience. We have somebody from China, from Japan, Australia, New Zealand, Germany, and somewhere else. Of course, it's very hard for us to find a time that fits everybody, but I feel very touched when people stayed up 3 a.m. waiting for our talk. Thank you. So all around the years, we provide all those seminars, talks, but one thing I want to remind everybody is our annual conference. This year is our 23rd annual conference, and we've been here for more than 2 decades. So 24 years. And then this is our 23rd annual conference. It will be held on October 2nd and October 3rd. I will just give you a very, very brief introduction or a quick peek about what we are going to talk, what we are going to do. So we have session 1, it's about frontline bioscience researchers. Like over the last year or two, everybody's talking about COVID-19, and sometimes you feel very tired of this topic, and you hope everything's just a pass. But we have researchers talking about not only COVID-19, we're talking about viruses, where they come from, where they go. So we have invited the world-class speakers talking about this subject, and also we have people who's working with genetics, talking about gene editing, and So for each session, we'll have 3 to 4 speakers. I hope you will find that it's interesting and come to our conference. And the second section is drug discovery and development. Actually, as of yesterday, we had a very, I think it's a very interesting topic about small molecule delivery to your body. It's like you send for example, mRNA, a vaccine. So this is a technique they're going to talk about, which I think has a big, bright future. And I think a lot of people will focus on it, no matter whether you're professor, scientist, or CEO, or you just want to invest your money into some place. That's a good one. And also we have new technologies and innovations. We have something called synthetic biology, and also optical genetics. Something is really new to me. And well, you probably hear it somewhere here and there, but as a big thing, it's, I think, inviting them to give a review or to have a vision of what's going to happen. I think it's very powerful. And for me, I think it's very interesting. And session 4, it's usually we put a very heavy weight on that each year for investment and the regulations. That's when you invest. So for investment, we always talk about what happened in the past few years, what is hot now. And the most important, I think, is you try to find out what will be hot in the next few years or next decades. So this is about our annual conferences. This is the website. I create a WeChat group. If you have any questions about today's talk, about Dr. Pan's talk, or about Chinese New Hope Cancer Care Foundation, or about the CBA, you can always go into there. And then this is a very temporary group. Once we exchange the information, or you have any questions, we answer. So that is for this purpose. Okay, now I have a very short, I try to make it very short about Dr. Pan, where he graduated from, what he's doing right now. But when I read all the introduction on the Kaiser Permanente Doctor's Dictionary, I find it's very hard. So I'm going to list some of it for you to see. Of course, he got the medical degree very early back in China, and then he moved it to US, got a PhD. So by looking at the PhD, the molecular and the cellular biology, you can understand this doctor is some doctor that's probably different than doctor. He probably have the inside view from a different point to understand the disease and the treatment. So I feel very lucky to have him here. And then he got postdoctoral fellowship and the residency in UCSF for us who is residing in Bay Area, of course, or maybe the whole world would know what UCSF is, right? And then oncology fellowship in Stanford University from 98 to 2002. And then he started work at Kaiser Permanente, specializing in soft tissue and bone sarcoma. From the discussion with Dr. Pan, I don't think his interest is limited in soft tissue and the bone sarcoma. You will see when he speaks for today's talk, and he has publications in cancer and stem cell research, and also now he's working on researching sarcoma and in other types of malignancy, as well as immunotherapy of cancer, the topic that we just had it 2 weeks ago by Dr. Mu. But today we have a different view to look at the immunotherapy. And also he got this list is just going on and on. So I try to push everything in. The only thing that I didn't put it here, I think I should, is he got a lot of awards from young scientists to research fellow, research, I should say, awarding for outstanding work. So I think by looking at this list, you would expect there's no limit for him to reach each corner where he wanted to speak. Okay, here, Dr. Pan, it's your time. Thank you.
Thank you, Su-Ming, for that kind introduction, and thank you everyone for taking your precious time to join us today. I'm gonna share my screen and then we'll get started. Let's see, how do do you wanna continue? Yes, so let me see. Okay, I got my PowerPoint here. So again, thank you everybody. You know, I should confess to you, you know, in my early days of practicing oncology, and I often got questions from patients about nutrition and asking me to refer them to a nutritionist. And I wasn't always very happy about doing it though, because, you know, in my mind we're thinking, you know, what's so sophisticated about nutrition? You know, when I was growing up, nutrition meant that having enough food to eat, right? But here in the US, with 1/3 of population, you know, adult population being obese, that nutrition really means having too much to eat. So, you know, everybody probably means different things when they talk about nutrition. And I wasn't always clear, you know, when people are talking about nutrition, what it exactly means. But, you know, in 2015, there were 2 publications in Science that really caught my interest. And one's here from a lab in France. The title is called Anti-Cancer Immunotherapy by CTLA blockade relies on the gut microbiota. And this paper really is summarized here. So I'm kind of fumbling on the PowerPoint. I'm gonna move the Zoom screen a little bit, and so I can see my slides. So, you know, what I found was that you know, when you create mouse model with, you know, tumor implants, that those mice that were treated with antibiotic or kept in a germ-free environment could not respond to CTLA blockade, you know, the immune checkpoint inhibitor. But when they fed the mice with a bacteria called the B. fragilis, or by immunize the mice with the polysaccharide from the B. fragilis, and they were able to restore the response to, you know, CTLA blockade, which is really ipilimumab we use in the clinic every day. And the other paper is from University of Chicago, and the title here is called Commensal Bifidobacterium promotes anti-tumor immunity and facilitates anti-PD-L1 efficacy. And I, you know, copy and paste one small piece of data from the paper. So, you know, when we give immunotherapy, or we call immune checkpoint inhibitor to a patient, on average, probably about 20–30% of the patients who respond. majority of patients still don't respond. It really varies from disease to disease, condition to condition. But then really the question is, why do some patients respond, why some patients don't? So then a few labs then realized that also by doing retrospective studies and combined with laboratory studies, I realized that some patients who took antibiotics, for example, when they having dental procedures, took antibiotics, before they started their immune checkpoint inhibitor, or during the treatment, they don't respond to the checkpoint inhibitor as well. And so really there was evidence, clinical evidence that if you treat a patient with antibiotics while they're receiving immune checkpoint inhibitor, you could really reduce the efficacy. And so, this study from University of Chicago look at the mechanism of that, and then they find that those patients who have deficiency of a bacteria called Bifidobacterium have low response rate. And when they take those mice model and treat them, fed them with the bacteria, and they were able to restore the response to the immune checkpoint inhibitor. And this piece of data really shows the interferon gamma production. Interferon gamma production is key to having a response to immune checkpoint inhibitor, right? And so after they fed the bacteria to the mice, they were able to tell the difference. So this is a fascinating experiment now they observe. Is one, like, TAC represents the mice from Jackson lab, called Taconic lab. JX represents mice from Jackson lab. And you can see that, different mice have different level of interferon gamma. And when they combine Jackson lab and Taconic labs mice together, the Jackson labs mice can enhance the interferon gamma level of the Taconic lab, right? And so really that tells us the gut microbiota has a lot to do with the response to immune checkpoint inhibitors. So, having explaining about those two papers, I thought perhaps we can come back to just some basics about microbiota. So you can see on this picture, this cows are grazing the grass, right? And then I put a line here. This is probably from Lu Shen, who in 1920s wrote this, and I'm not even sure, what essay this line was from. I copied from somewhere. It says, "I'm like a cow who eats grass and make milk." So it sounds like cow eats grass, but can make milk for human. It's pretty incredible, right? But if you think about it, human can do that too, just human cannot eat the grass. So for the grass, for those cows grazing the grass, really they're eating two things. One is prebiotics, the fiber, and the fiber from those grass was really cellulose. And another is bacteria, we call probiotics, right? And this is the picture, a bunch of different kind of bacteria. So you see that they really were grazing prebiotics and probiotics. And cow has 4 stomachs, and they have all kinds of bacteria that can produce all kinds of enzymes that can digest cellulose, which cellulose is basically sheets of polysaccharide, which is basically the glucose that's linked together. It is a sheet, and you need a very powerful enzyme like glycoside hydrolase or polysaccharide lyase to break down those cellular bonds, the molecular bonds. And so those could be digested and absorbed, right? But we human cannot really eat the grass. I guess 50,000 years ago, when humans were not cooking, eating grass was probably okay. You probably could digest them. But now, we cook them. We cook vegetable to eat them. And the difference is that when you cook vegetable, the vegetable contains the kind of fiber that is shorter, simpler. And one kind of fiber is called Inulin, and which you can see that we human make the fiber into powder, and you can actually take the powder and just add some water and drink it. And you can see this molecule here is a lot simpler than cellulose, and it's a lot easier to digest by the human stomach because human only has one stomach, right? And here I just wanted to give you an example of certain vegetables that contain high content of inulin, like leek, onion, and rye, and so on. But in terms of probiotics, we humans don't just graze grass, and then when cow grazes on grass, they were also eating soil. The roots, they eat the roots as well. And the soil contains very abundant bacteria. So the cow really was eating probiotics as well. Now we humans don't graze grass, so we get probiotics from certain food that we prepare, like kimchi, that contains those very important probiotic. And yogurt, right? I guess most of the people eat them regularly, or at least from time to time in pickles, called pao cai. Also, every time when I get a sandwich, the first thing I eat is pickles, really. And all those three different kind of food are fermented vegetables and contain abundant bacteria or good bacteria or probiotics. So let's just touch on some basics about gut microbiota. Now, if you think about it, human gastrointestinal tract covers a huge surface, and this is about 2,500 feet to 4,000 square feet, which is bigger than my house actually. So you can think about it. We have such a huge gastrointestinal tract, and over our lifetime, on average, 60 tons of food pass through our gastrointestinal tract. And then our gastrointestinal tract contains about 100 billion bacteria, archaea, eukarya, and viruses, right? And probably more than 2,000 or even 3,000 different species of bacteria, and they could be categorized into 12 different phyla, and this was done by called the Human Microbiome Project. And basically 4 different bacterial phyla, like this one, Proteobacteria, Bacteroidetes, Firmicutes, Actinobacteria. Those 4 phyla consist of 93 of the bacteria in our gut. And those bacteria do these things. I'm sure I have this, the Zoom screen cover my screen a little bit. But I hope you can see the whole screen that, this fiber, those vegetables that are digested by the gut microbiota through fermentation into those molecules you can see, and then they into we call tricyclic cycle, and through the other coenzymes here, then they produce we call short chain fatty acid, and one is called butyrate, another is called acetate, and another is called propionate. Right? Now, those are very important short chain fatty acids. They do a lot of things. They can passively diffuse into the endothelial cells of the gut. They can also be actively transported into the endothelial cells. Now, they can also bind to G protein coupled receptors on the surface of many different cells, including gut endothelium, immune cells like dendritic cells, like T cells, B cells, and macrophages. And by doing so, regulate immune system. And so this is a diagram about those 3 very important, we call short chain fatty acids. And then, you know, so just a caution that, you know, if you don't regularly eat a lot of fiber or, you know, yogurt, and then suddenly you begin to eat, you know, a lot of yogurt and, you know, pickles or kimchi or, you know, those stuff, you may actually get a lot of gas, because you can see this acetic acid can be actually converted into methane and CO2, and you can get a lot of gas. And so, you know, when you start doing that, you may want to be careful. But over time, then your bowel get used to it, and then that will, you know, get better and better. So what do those short chain fatty acids do in our gut? One is that they provide energy for the gut endothelial cells. So our gut endothelial cells don't really use a lot of glucose. They actually take up those short chain fatty acids for energy. Short chain fatty acids, like acetic acid, can also stimulate, you know, B cells to produce reactive IgA to neutralize pathogens like E. coli, like C. diff. And they can also reduce inflammation of the gut endothelium by inducing, you know, Tregs, regulatory T cells, and modulating cytokine production in the gut. They can also stimulate production of the beta defensins, which are, you know, small polypeptides that can neutralize viruses and bacteria. So they really play, you know, many important roles in our gut. Let's see. So this is a very interesting slide that I took from a Nature paper. Really came from, you know, Wang Jin slide, a BGI Shenzhen. And they did a great study by sequencing the microbiota 124 European people, you know, by collaborating with IMBO Lab in Germany and in many other countries. And they were able to identify the bacteria species in healthy people. You can see here in the quadrant here, PC1, but patients who had Crohn's disease or ulcerative colitis, their bacteria, you know, diversity or abundance are a lot less than the healthy people. And that was really interesting because, you know, Crohn's disease and ulcerative colitis are really inflammatory bowel disease, right? So that really says something about the importance of the microbiota in bowel inflammation. So then the question is that, you know, we're talking about cancer today. What does diet have to do with cancer? And I'm sure, you know, you ask anyone can tell you something about it. So one is obesity. You know, people who put in a lot of calories, get really obese, have increased the risk endometrial cancer, breast cancer, biliary cancer, even colorectal cancer. And there's very good evidence about it. Now, say they don't eat a lot, but they don't eat the right diet. A lot of people eat high-fat diet, you know, a lot of saturated high-fat diet. And there's really good evidence that, you know, low-fiber, high-fat diet really increases the risk of colon cancer, right? You know, when people eat a lot of saturated high-fat diet, most of those high-fat diet really contain, you know, three types of we call long-chain saturated fatty acids, like, you know, myristic acid, stearic acid, you know, palmitic acid that contains, you know, 14 to 18 carbon fatty acids. And those are very difficult to break down, you know, their molecular bonds are very rigid. But then the question is, you know, how do high-fat diets cause cancer? What are the mechanisms of them? Now there have been, you know, of course, tons of studies about the mechanisms of high-fat diet causing cancer. I wanted to just describe a couple of experiments that are really fascinating. But you know, as a summary here, the high-fat diet increases bile acid output in the liver, right? And it increases the bile acid concentration in the gut, and which actually is not good. Another is that high-fat diet increases inflammation of the gut endothelium, and high-fat diet causes microbiota imbalance in the gut, which could lead to increased cancer-associated bacterial biofilms, right? So what is bacterial biofilms? Bacterial biofilms really means that you have, a sheet, sheets of, bacteria that aggregate and wrapped up by polymer like mucus in the gut. And those aggregates of bacteria may get in touch, get in contact with the endothelial cells of the gut. And when it happens, then they can directly invade the gut endothelium. They can even get into the deep tissue, and it can cause inflammation. And then with many other mechanisms, that can cause cancer. And this is a very nice study published in PNAS a few years ago that shows that, when they look at the patients with cancer, colorectal cancer, and they find that those patients, which we call right-sided colon cancer. So you see right-sided colon cancer, it's different than the left-sided colon cancer, right? And so right-sided colon cancer really stands for cancer from the cecum all the way to the transverse colon. And the left-sided colon cancer is from the splenic fracture down to the rectum, right? But what they found was that only in the right-sided colon that they found bacterial biofilms. Those bacterial biofilms or aggregates could be found either on the site of cancer and could be found inside the cancer or could be away from the cancer. But they found very few, you see those red dots. Those are all the location of the bacterial biofilms. Now, but on the left side colon cancer, they found just a couple of them. So the difference was somewhere around 85 to 13% difference. Now, right side colon cancer has worse prognosis than left side colon cancer. Now, the mechanism of that difference is still not clear. Could the microbiota difference in two different types of cancer be responsible for that? It's very, very likely. So this is very fascinating. Now, how? So the next study is about high-fat diet. How does it cause cancer? By regulating cancer stem cell proliferation. And this is a Cell paper published a couple of years ago from Ronald Evans' lab. And now this is a diagram on the metabolism of cholesterol to bile acids. You can see that when we eat a fatty diet, that basically contains a lot of cholesterol. And you can see here that carbon 27 cholesterol is metabolized into the cholic acid or deoxycholic acid, right, through gut flora. And actually those metabolites from the bile acids could be toxic to our gut endothelium when the concentration is too high. And I copy and paste this diagram from the Cell, this graphic illustration of the study's main points. So what they did was that they took the mice we call APC min mice, and those mice really have the deletion of the APC gene. So you all know APC gene is a gene responsible for familial polyposis colorectal cancer, right? And people who have germline deletion or mutation of APC gene have 80% probability of developing colorectal cancer in their lifetime. Now, mice or people just have the APC gene mutation, they don't develop cancer. They have the other factors that compound for the gut to develop cancer. And one factor here, in the experiments that they use high-fat diet. So high-fat diet on top of the APC mutation increases the bile acid secretion and promote cancer stem cell proliferation and can induce DNA damage. See, now, interestingly, when they use a agonist called FXR agonist, now what happens is that FXR is a nuclear receptor. Bile acid metabolites will inhibit FXR, and by inhibiting FXR, they can inhibit a lot of physiological process regulated by FXR, and cause the proliferation of cancer stem cells and DNA damage. But when they treat the mice with FXR agonists, they find that they could actually inhibit cancer stem cell proliferation. They can protect the endothelium from DNA damages, and they can change the bile acid composition and levels in the gut and prevent cancer progression. And so I took one piece of the data from their paper, which is quite fascinating. You can see by treating the mouse model with FXR agonist called FATX can improve mouse survival. You can see on this one, this graph here and this graph here, the black curve are the control mice. The right curves are the mice that were fed with this FXR agonist, FAXD. And you can see the mice live a lot longer, almost 50 weeks compared to 30 weeks here, right? Now, interestingly, when they sequence patients, when they take patients and do RNA sequencing, they could separate the patients with low and high risk into different gene expression profile. And what they found was that those patients who had low risk signature had a signature that was similar to what they saw in the mice treated with FXR agonist. But those patients who had high risk had a signature that was similar to those patients who were fed just with the high-fat diet. So this is quite fascinating, that you can actually modulate the bile acid metabolite, and by doing so, possibly prevent or treat colorectal cancer. Now, this is another paper from MIT by David Saptini's lab, published a few years ago in Nature. And you can see that, this on the left side, this is the mice that were given the control, the good regular nutrients. On the right-hand side, the mice were given high-fat diet. HFD stands for high-fat diets. And you can see that when the mice were fed with a high-fat diet, those lipids were stimulated, called part delta. Part is basically peroxisome proliferator activated receptor delta. Now, this is a very important pathway, and this pathway has been targeted, by drug companies to make drugs for treating diabetes. So there are several drugs for treating diabetes that have been, in clinical use for many years that are actually part delta inhibitors. Now, high-fat diet stimulates part delta, and part delta then stimulates beta-catenin activity. And we all know that beta-catenin is a very critical component of the Wnt pathway, right? Now, stem cells, the gut endothelial stem cells are regulated by the Wnt pathways. So when beta-catenin activity is increased, you can imagine that the endothelial stem cells would have increased the population and perhaps their mitosis will become dysregulated, and then they will develop mutations and develop into tumor cells here, right? So those couple of studies were so fascinating. Now I wanted to just come back and just briefly touch on what are some of the good bacteria and what are some of the bad bacteria we encounter a lot in our daily life. For the good bacteria, you can see lactobacillus. Is a bacteria we eat in yogurt, and also importantly, is the most important a good bacteria in the vagina of women, because when babies are delivered, they pass through the birth canal and they get some of those bacteria, and that's very important for them to get popularized in your gut. And so they develop the good microbiota. Those babies were delivered by C-section up until now I have the benefit of that. And so there's, you probably all know that the babies who were delivered by C-section may have increased the risk of infection. And then we also, encounter a lot of other good bacteria, like, Ficariobacterium, bifidobacteria, which we mentioned earlier, and also some species of Bacteroides, like B. fragilis, that can repair colon weakness. Some of the bad bacteria here, E. coli, we all know E. coli is responsible for a lot of bad infections, like urinary tract infection, H. pylori, you all know about that. There's one called Fusobacterium nucleata. C. diff is a very bad bacteria. Now, how do bad bacteria cause cancer? And here I want to just give you an example of the mechanism of how a bad bacterium can cause cancer. Like, for example, like this one, Fusobacterium nucleatum, has this factor called fat A, which could bind to beta-catenin. And then beta-catenin is a very important tumor suppressor gene, on the surface of the cells, right? Now, fat A can also inhibit the innate immunity by targeting toll-like receptor 4 in MYD88. So MYD88 is a vector for the innate immunity signal transduction. So when those proteins are inhibited, the innate immunity is also inhibited. Now, and another protein from this bacterium called FAT2 can bind to a molecule called TIGIT on NK cells to drive immune evasion of the tumor cells. They can also inhibit, this molecule can also inhibit autophagy by targeting, a couple of microRNAs we call MER18A and 4082. So they can target a lot of pathways in human gut to cause cancer, right? So, I wanted to transition to talk about the things that we possibly can do by understanding how gut microbiota impact human. And by studying mice and human, we can understand a lot about the impact of gut microbiota. And this is a fascinating study from Washington University by Jeffrey Gordon's lab. So what they did was that they took twins discordant for obesity and see how they modulate gut microbiota in mice. And this is how they did it. They took 4 pairs of twins. So twins one twin had a BMI, if you look at the BMI here, one had a BMI of 23, another had a BMI of 32. Another twin pair, one had a BMI of 25.5, another had 31. And another pair, the BMI is 19.5, and the obese one, it was 30.7. And another pair, one had a BMI of 24 and another had a BMI of 33. So 4 pairs of twins one has the perfect BMI and another is obese. So when they look at the bacteria diversity in their, in their stool, you can see the differences. You can see the lean donor, the lean twins here, they have high bacteria diversity. You can see here, right? The obese twins here, they have low bacteria diversity. That's really interesting, right? When they also measure the body composition, you can see that their what they did was that they fed we call non-biotic mice, which really means germ-free mice, right? The germ-free mice do not have any germ in their gut, and they're kept in a germ-free environment. So they will feed those mice with the feces from the different twin, right? For example, when they fed the mice with the lean, called the lean twins, right? Their body, the mice's body composition is here, very low. But then they fight the germ-free mice with the obese twins feces. You can see their body mass is a lot higher. And then when they also measure the fat mass, they see basically similar data. The obese twins feces make the germ-free mice obese. And you can see here is by time course. You can see the blue line is the change in fat mass over time by the mice that were fed with the obese twins feces. And the right line, lower line here are the mice that were fed with the lean twins feces. And you can see the difference. So that's quite astounding. You can actually replicate this difference in mice just by feeding the mice with the twins stool. Quite amazing. And then they sequence the feces from the mice and see the changes of the microbiota. When they put the mice together, we call cohousing the obese and the lean mice, right? And see what happens because when you put the mice together, when you put even the dogs together in one cage, they eat each other's the food. Sometimes they eat each other's feces, right? And so you can see that for on the left-hand side, the microbiota profile here is from the lean twins fed mice. And you can see they're pretty similar. Before cohousing is here, right? After cohousing, the microbiota profile looked very similar. But if you look at the right-hand side here in the middle, like this zone is so empty. And this is from the mice that were fed with the obese twins feces prior to cohousing. So they were kept in a germ-free environment by themselves. And then, but after they kept this mice with the lean mice together, cohousing them, and so what happens? You can see the changes in the microbiota. So it's very similar to the microbiota from the lean mice on the right-hand side. So you can see just by cohousing them, you can change the microbiota of the obese mice, which is quite interesting. And then, this is even more interesting than that, though. What they did was that they measured the called short chain fatty acid of the cecum, right? They, sacrificed to the cecum and measured the concentration of the short chain fatty acid profile. And they measure propionate, butyrate concentration of the obese and lean mice. They were cohousing. So the control here is the obese mice, right? They were kept alone. You can see the propionate is low, below 4 here, right? And here in butyrate, you can also see it's very low. But the lean mice, they don't need to be cohoused, just lean mice. They have very good level of propionate and butyrate here. But when those obese mice were co-housed with the lean mice, their level of propionate and butyrate dramatically increased, right? And almost to the level of the lean mice by themselves. So this, this study really confirms that not only there is a change on the microbiota, it also led to the physiological changes of the gut endothelium, right? And then Jeffrey Gordon's lab, it recently, probably just 2 or 3 weeks ago, published another paper in Nature, and it did another really exciting study by evaluating microbeam-directed fiber snacks in non-probiotic mice and humans. So what it did was, I took this a picture from this from Science as a commentary, and I thought this picture will help us to illustrate the study's interesting results. So on the upper hand, you can see this is, the experiment on mice. On the lower part, this is the study on human. So on the upper part, you see that they would, feed the mice, germ-free mice with high-fat diet, and they took the feces from the obese people and fed the germ-free mice, and so the germ-free mice fed with the obese people's feces will become obese, right? And so what they did was that they would treat those obese mice with, they call it designer fiber meals. You see on the title here, one is called pea fiber supplement, right? Another is called orange fiber supplement. Another is called barley bran supplement. You all know those supplements, very good fibers we human eat. We probably could get it from, the Costco or Safeway. And finally, and then look at their microbiota. And so what they found is that if those obese mice are fed with those fiber almonds, fiber meals, then they have more fiber degrading gut bacteria. So they discovered that those mice actually have increased bacterial diversity. They have more bacteria that can degrade the fiber, which is very important. If you cannot degrade fiber, then whatever fiber you eat will just pass through and will not be, broken down into, nutrition, right? We call, nutrients to be absorbed by the gut endothelium into the body. Now the lower part, they actually took a human and then randomized them into 2 groups. The obese people here will be given just the high-fat, low-fiber diet like they normally eat. Some people just like high-fat diet and low-fiber diet, and they could never change. Another group of the people, the obese people, will be given, Inulin supplement. You probably still remember in the, early slides, I showed the Inulin supplements, right? And then they would check the feces from the 2 groups of the obese people and look at their microbiota. And so what they found was that the obese people, when they were fed with Inulin supplement, had increased bacteria diversity, significantly more than those obese people who were just given the regular high-fat, low-fiber diet. And this is really interesting. This really means that you can actually change people's diet and change their microbiota profile. And by changing their microbiota profile, you can change their health, right? And so we're no longer just talking about, yes, or yeah, if high fiber, everybody is talking about organic food, high fiber food. When you go to Costco or Safeway or somewhere, you want to buy organic, whole grain fibers, right? But this is really an experiment that really shows that it could make a difference in terms of altering our bacterial profile in the gut in the physiological process. So, with understanding not a lot more than a few years ago about our gut microbiota, then a lot of companies actually, probably here in the US and in China, are starting to look at the microbiota as therapeutics and see if they can come up with a pill that can just fit human. And now cause a lot of pills over the counter. They're called probiotics, or different kinds of labels. You can get it from anywhere called probiotics, and you can take the pills, and of course the label will tell you how great those probiotics are. And there's no study that really could show any of those probiotics are actually useful, or if they are any more useful than just eating a cup of yogurt, or just eating maybe a dish of kimchi or pickle, right? But if we know enough about gut microbiota, perhaps we could do something really to alter the human gut microbiota and to improve human health, and perhaps to even alter the treatment for patients who are not able to respond to immune checkpoint inhibitor, right? Like, in the early on when I showed a couple of studies that were published in 2015. Now here is the paper I copied just a commentary from Science, and this was published in 2018. And the commentary was that good bacteria help fight cancer. And so what I found was that those patients who had lung cancer and kidney cancer, when they looked at feces, they had low level of fecal called Akkermansia mucin, that particular bacteria. And so they did create a mice model, and then fed the mice with this particular kind of bacterium, a mucin. And then what they found was that when they fed the mice with this particular bacterium, they were able to restore the response of the mice to PD-1 blockade, which means drugs like pembrolizumab, nivolumab, that we use every day in the clinic. All right. So, of course, this is still in mice, in experimental, not human, but eventually, if we can come up with really a pill that can do what we are able to do in mice, it will be quite fascinating. We can really improve the efficacy of immune checkpoint inhibitors, a great deal. Well, there are studies now that have been done ongoing, really can demonstrate that using microbiota is a therapeutic. And this is a study that was also published in Science Translational Medicine that showed that they can feed those patients who had to go through allogeneic bone marrow transplant. And let's say they had leukemia, they had to receive allogeneic bone marrow transplant. When they receive allogeneic bone marrow transplant, they have to be given a lot of antibiotics to protect them from infection. But when they were given a lot of antibiotics to protect them from infection, their gut microbiota is basically wiped out. All those antibiotics just kill good or bad bacteria altogether. And this study showed that they would take the feces from those patients before the bone marrow transplant and freeze them. And then after the patients were done with the allogeneic bone marrow transplant, and then they will fed their own, the feces, the frozen feces, and then they will feed them back. And they found that by doing so, they were actually able to improve the outcomes of those patients. Those patients recover faster. They probably have lower mortality, and that's complications. So that was quite fascinating, but I wanted to end here by just summarizing what I have really described in the talk. And then I will end the talk and see if there are any questions. So in summary, really diet is important in supporting gut microbiota, right? The bacteria in our gut will not eat meat. They are vegetarian. They will eat fiber. So you have to supply them with fiber. And so they will be able to convert the fiber into short chain fatty acids that are very critical for the health of the gut endothelium and the immune system. Microbiota can produce, key nutritional ingredients for gut and body, which, the main components are really the short chain fatty acid I just mentioned it. And, with imbalance of gut microbiota, there will be increased risk of bowel inflammation, and chronic bowel inflammation can cause cancer. We know that patients with Crohn's disease or ulcerative colitis, those two inflammatory bowel diseases have 30 times higher risk of developing colorectal cancer than regular people. And another piece of information is that high-fat diet really can cause bowel inflammation, and can lead to increase the risk of colorectal cancer. Now, high-fiber diet can correct the gut microbiota and can also correct the serum metabolic profiles, which I forgot to describe in Jeffrey Golden's study called the Designer Fiber Meals. They also measure the serum metabolites, and they were able to confirm that not only the high fiber diet were able to change the gut microbiota, they were able to change the metabolites in the serum, and which is so associated with good health. So that's my talk today, and thank you very much for taking the time to join us. And if you have any questions, I'll be happy to do my best to answer any questions. Thank you.
Thank you so much, Dr. Pan. You really opened a big window for us. And the majority of the time, or most of the time, I attend nutrition and seminars or other doctors talk about nutrition, about microbiota. A lot of times there are yes, no, or maybe so answers. So today you just really made a very strong statement. It is related to cancer, related to our health. Thank you so much for those fascinating studies that you showed us. Of course, maybe not yours. You made it very clear. You borrowed from someone else, but thank you for borrowing it and show us. I really learned so much. I believe the audience also learned so much. They sent me a lot of questions, and I know in the chat box, if you have questions, you can still submit. And I also got questions from my own friends group. So I would like to ask you something. We started with the diet. We mentioned that the probiotics is good, but do you suggest people that just taking probiotics pills or powders or whatever formats as a daily intake?
So that's a very good question. The problem with taking probiotic pills is that we really don't know how much can actually survive the acidic environment of the stomach. The pH in our stomach is as low as 1.5. So our stomach is very, very acidic. So when you take the probiotic pills or powder or whatever, that how much of those you take actually survive and get to the colon? It's not clear. So you eat yogurt. That it's a good nutritional food. How much does yogurt pass on the good bacteria to the gut is also not clear. But for me, I eat yogurt every morning, and that's all I eat is the breakfast.
You look very healthy, so I think I think the thing is thinking about the stomach acidity is so high. So the point is probably whatever you put it, you eat, you intake it, and then passing this stomach process maybe change the property of the bacteria, maybe killed a lot. So what you mentioned also another point, the prebiotic, the food for probiotics, right? So
right.
if you take something that make your body to increase the production or increase the amount of probiotics, is that the way we should go? Or instead of just tons, like the product you see on their commercial always say it's millions, billions of probiotics. So instead of that, we should just let our bodies work.
Yeah, perhaps a combination of fiber and sources of probiotics is probably the best thing to do. Of course, nobody can point you to experimental evidence about that. But I think just by intuition and by the evidence we have from human and mice, I would say that good amount of fiber and combined with some probiotics every day, different kind of probiotics. Like I like kimchi. I eat kimchi from time to time. That's just as good.
So talking about the kimchi is something that had the process of it's a fermented veggie, right?
Right.
And also thinking about some Chinese food or some food that we add salt, sugar, vinegar, and then after a couple of days, it's as good. The taste is as good as the pickles. So what's the difference between these two? Which one's good and which one's not good?
Well, I don't think we know. You go to different store, you get different kimchi, and they taste different. Costco has kimchi, HMO has kimchi.
So what we are looking at is, as long as this is fermented, it may bring you a lot of bacteria. But for the other part, like you put salt in veggie a few days, maybe it's not fermented. Probably that's not going to help.
Right. So, fermented food is different than preserved food, right? Like in China, people eat a lot of preserved vegetable, which increases the risk of, esophageal cancer, stomach cancer. People who in Hong Kong, Guangdong, eat a lot of preserved meat and fish, and that contains a lot of nitrates that are associated with increased risk of nasopharyngeal cancer, stomach cancer, and so on.
That's a very good answer. Yeah.
So there's some questions on the chat here, if you don't mind, that I could take a look and see if I can address the questions. One is that, let's see, my window is pretty small here.
I would go to that place later. You're not so easily get away from me.
Okay. Why don't you go ahead and then maybe you can.
I did make some notes while I was listening to you. And you mentioned the continuous feeding versus the restrictive feeding.
Oh, so I didn't get to feeding, that. Right.
Yeah. So for cancer patients, when they are during chemo time, the doctor always says that eat a smaller portion with more frequency. So in that time, we don't think about whether it's good or not, but smaller portion and the more frequent is better.
So that's no different about, that's different. When we say eat small amount and eat it more frequently, we're talking about, patients who sick. They've got cancer, they're probably on chemotherapy, they're probably not able to eat a large meal every time. They can get full easily. But in order to keep up with, nutrition and calorie sources, they need to eat more frequently, right? So that's the advice we give to a lot of patients who have, trouble keeping up with calories. But when we talk about continuous feeding or intermittent fasting, we're talking about when someone likes to eat all the time, and so they may be eating snacks from morning to dawn to dusk, and they may feel like I'm not eating a lot. Some people say I drink 6 cups of juice. That's all. But if you keep feeding, you do not give your liver time to rest. liver is a huge, metabolic organ. And when you do intermittent fasting, you give your liver, your body a rest. If you think about it, your bowel needs a rest, right? So if you keep putting stuff down to your bowel never gets a break. Now, we human need a break. We need to sleep. But how about our bowel? How about our liver? So every organ in our body has this called a circadian rhythm. At night, they need to sleep, need to rest. But if you keep feeding them, let's say at night, there's very good evidence, epidemiological evidence that shows that people who work night shift for a long time, like 20, 30 years, have increased the risk of breast cancer and prostate cancer. And that is likely because, when they work night shift, they have to eat at night, right? But, the liver is supposed to rest at night. And so when you eat a lot, the liver gets tired. They cannot metabolize those hormones like, estrogen, like testosterone. So those people have constantly higher level of the hormone in their circulation, and that increases the risk of breast cancer, prostate cancer, right? But when you give your body a rest, you eat, and you are very disciplined that for certain hours you don't eat, the body has a chance to digest the food that was given. And then also body has a mechanism, which, I did not discuss today, called autophagy, right? Autophagy, let me see, I have the slide here if you can see it. Really, it's a physiological process. Every single cell has this autophagy process that can recycle the materials from the degradation of cellular organelles. All the cells in the body have to die. When they die, they break down, and those broken down materials could be recycled and reused to build new cells, new organelles, right? But if you keep feeding your body, then your body would not have the chance to recycle those degraded materials, and that could lead to physiological disturbances.
Thank you so much for such an in-depth explanation and about why we shouldn't continuously eating. And also, I noticed one slide you mentioned to avoid antibiotics or avoid the steroids when you have treatment. So,
right, I removed that. I thought that during the talk, it was kind of clear without having to prepare a separate slide.
I see.
There's good evidence. Now we would tell the patients, and this is essentially on a daily practice. You have patients who are immune checkpoint inhibitor, like pembrolizumab, like Keytruda, and they have a dental abscess. They need to have the tooth removed. They need to be given a course antibiotic, and if that cannot be avoided, They have to take a week or 10 days antibiotic, or say, yeah, go ahead and do that. And I'll tell them to eat yogurt every day. Say, when you take an antibiotic, I want you to eat yogurt every day, at least 1 or 2 yogurt a day. And that could be helpful. Some patients really see the benefit of it. Antibiotic can cause diarrhea. When you eat a yogurt or 2 a day, you probably won't develop diarrhea. You probably will tolerate the antibiotic a lot better. Right? Steroid is bad when you going through immunotherapy. And the reason it's bad is because the steroid can cause what we call apoptosis of the immune cells, like B cells, like T cells, right? And so that certainly can compromise, patients' immunity when we're trying to augment their immunity.
So this is because we had a talk 2 weeks ago about checkpoint inhibitors mediated side effect. So where we had a lot of inflammation in different sites, colitis, pneumonitis, all those things.
Right.
Doctor always use steroids at the end when there's no way to do it. So do you suggest, maybe this is a little off topic, do you suggest when you use steroids, you stop the inhibitor, or you can still keep it on?
Well, no, so the problem is that when the patient develops serious side effect from immune checkpoint inhibitor, like severe diarrhea, like epidulumab does that a lot, And you have to stop using the drug, epiduvumab. You have to stop using it, and you have to give them steroid to stop the inflammation. Otherwise, patients can die from severe diarrhea. You know, occasionally you see a patient who develops we call it interstitial pneumonitis, and you have to stop because some patients really can die from that type of serious side effect. Not only you have to give steroid for some patients who do not respond to high dose steroid, you have to give something like, TGF alpha inhibitor to really suppress the immune system to reverse the toxicities. So it's not contradictory. It's just like you have to treat the side effect. When you have to treat the side effect, you have to use all the tools you have.
Yeah. I think doctors are just like detectives to find out what that is and save life first, and then we talk about the treatment of cancer, I think.
Right.
Yeah. So there's also one thing, a few studies that's, I think, very interesting, almost made me think, if we can introduce cows digest the gut microbiota into human, do you think we can eat grass and produce milk? Of course not. So my question would be, when we do the experiment to introduce somebody or someone somewhere's fecal transplantation,
right.
How do we do it?
Oh, you put I call it a feeding tube, NG tube through their nose and all the way down to the small bowel. And for some patients, they don't want to know it. You just give them medicine and let them go sleep. And then you put those stuff, you put those frozen feces and dilute them into liquid, and then you just get a syringe and push through the tube.
Oh, I thought you push through the tube will end in stomach, but it's very smart. You put it into the small.
No, you put through the stomach, go to the jugular.
Then we'll kill it. Okay.
Yeah.
And how do you choose such a candidate? Like one patient needed this transplantation, and then the other one is healthy, good enough. The fecal contents is good enough. How do you choose that person?
Well, you choose a person who is in perfect health. And ask them, is it for donor, you want to know that that person is, completely healthy, and of course, patient needs to consent to it. We don't do that to our cancer patients. That's still in an experimental stage, but we do that for patients who develop called C. diff. Some patients who were given antibiotics, and this is very common. They develop C. diff. Basically, all the other bacteria are killed except C. diff is still there. And so you really need to replete your gut microbiota. And sometimes the only way to treat the resistant C. diff is to, feed them with microbiota, which is, now, there's one thing, there's one fact, and maybe you'll be interested in knowing, is that 40% of the dry weight of our stools are actually bacteria. 40% of them, that's a lot.
Yes,
right?
It is. Well, it is. Yeah. So there's no way we can have a test to test if somebody's fecal, the contents is good or not. There's no way.
You could do that, but I'm not sure if it's commercially available.
I see. Yeah, I think a lot of people in audience, they would know if the gut microbiota in my body is good or not, to see the balance.
I wonder, maybe there are companies working on that already. Otherwise, it sounds like it's going to be a good business. There's a lot of interest here. It's actually not difficult to do. What you do is take the feces, and then you do sequencing. You just get Illumina in a sequence called a 16S ribosomal RNA, and you get a profile.
Okay. I almost well, I have a lot of questions, but I will give the audience a little time. Let's go to the chat box.
Okay.
Will you be able to see it, or do you want me to read it?
I can see it, but I don't know where to start.
Okay, I will start. It's a long list. And one question is about mushroom. Mushroom is one of the fungus.
Mushroom is called eukarya. It's good. Mushroom is good.
So the question is if linzhi is good.
Well, just as good as anything else.
Okay, it's not
any better. I always tell patients, if we got, extra money, sure. Your money is it's a precious commodity, and you don't have enough of it. You probably don't need to spend $100 for a bottle. Then, that kind of mushroom powder, you could just eat any mushroom.
Okay, next one would be for you, Dr. Pan, would you allow me to study this lecture as patient care again? I'm not quite sure what that is, but.
Yeah, I don't know that question, what that question
is. I just want to tell you, any audience, if you have any questions later, I will have the code, a temporary group there. You can post your question over there, or you can post any other requests or question, so we can take care of it. And next is, what kind of digestive probiotic pills that you recommend? I think you already answered that question.
Right, yeah.
Yeah, so let's skip that one. What do you mean by repair colon likeness?
When there's inflammation in the colon, that the endothelium will be leaky. So that's, when they say, when you have diarrhea, right? We all do once for a while. And then there's inflammation in the colon, and sometimes you see mucus. Why? Because there's inflammation there. Then the endothelium is oozing out a little bit of the mucus, and so a good amount of fiber, good microbiota can help you to repair the leakness.
Perfect. And also, I think the leakness comes from the cell-cell junction. Once they open, then not only the small molecules, the large molecules also get transported somewhere they shouldn't be. Next one.
Basically, when let's say repair the leaknesses, is that the endothelial stem cells were able to renew, right? In the gut, endothelial stem cells are hiding in the niche, in the we call crypt, in the bottom of the endothelium. Right, they're hiding there. So when they need it, they begin to renew the make new cells, and those cells will migrate from the we call niche, from the bottom to the surface, and repair the leaky area.
Oh, okay, thank you. And he mentioned, can you share the slides? I'm not sure which one that one is. Do you know which one that is?
Which slide?
About the colon. Repair colon leakiness.
I don't have a slide. I just mentioned it.
Okay. Yeah. So maybe he meant something else. Just yeah. Right. Okay. And next, what kind of cancer from chronic bowel inflammation? So the question
kind of cancer?
Probably the question is bowel inflammation will cause what kind of cancer?
What kind of cancer? Well, I mean, all kind of colorectal cancer, right? It could be what we call right-sided colon cancer, probably could be left-sided colon cancer. It really depends what kind of inflammation that is, right? For for patients with right colon cancer, they tend to have a lot more immune infiltrates in the tumor, and for the patients with left-sided colon cancer, the tumors tend to be cold, which means that they don't have a lot of immune infiltrates. I'm not sure if that answers the question.
I see. I think maybe he meant, of course, we can understand that the digestive cancer, system cancer may be caused by bowel inflammation. I had one small question would be, would this inflammation cause, say, brain cancer, breast cancer somewhere else? We don't know.
No, that information is that wouldn't be directly associated. The information about would not be directly associated with breast cancer or other cancer. It will be associated indirectly with something like high-fat diet causes breast cancer, right? Does it cause breast cancer through the bowel? We don't know. Maybe there are different mechanisms that we are still not clear.
Yeah. The whole human body is all connected to each other. So never know what that is. Next question, right. I think we can still skip. It's about which one probiotic drinks or yogurt you recommend. And this is very interesting.
I don't eat the different kinds of yogurt, and there's no yogurt that anybody knows is better than the others. So get whatever you like and eat yogurt that's not as sugary. Now, some yogurts are very sweet. They add a lot of sugar. They add a lot of fruit in it. I normally try. I like a little bit of fruit in it, but I don't like very sugary stuff.
Mm-hmm. So that's related to low sugar diet. I think we should consider about that when you buy it. It's very interesting. We finally have some scientific evidence that changing patients' diets can help. Hope this will push more hospitals and provide a nutritional service to patients. I think information is very powerful nowadays. Like today, your talk definitely helped us to understand the deep inside what really is. When we talk about an acid, your body is acid, that's why you have cancer. And then you drink a lot of alkaline water, do a lot of things. Right. After this talk, so I don't think that will help so much.
Right, I mentioned here, there's a question here, Su-Ming, what do you think about kefir? I think kefir is okay to eat. And you mentioned about drinking alkaline water. I have been asked by a lot of patients about that. Basically, if you think about our bodies, the pH of our circulation, when you do a pH, what will be the pH you expect? Will be somewhere between 7.35 to 7.45. So there's a very, very narrow pH range in our body, in the blood circulation, right? So if you wanted to drink a lot of alkaline water to change your body's pH, that's impossible. You will have to drink a lot of it. And so some people read online saying, wow, I got cancer, I'm gonna, because cancer is acidic, I'm gonna drink a lot of alkaline water to make cancer less acidic. That's not gonna happen. You will not be able to drink enough to make that happen.
Yeah, that's people have, they thought they have evidence that once you drink a lot of, take a lot of alkaline sort of food, your urine will turn, the pH will turn higher. And then I did some small research on that because you have so much extra that your kidney just like you have a high contents of potassium, just to push them in the urine. So that's not relevant to what you're eating, right? I mean, for your whole body to turn but pH.
What urine pH changes a lot. It continues from 5 to 9 easily, but it doesn't reflect any changes in the body.
Okay, let's see. Are you ahead of me? I'm on 11:28 a.m. Oh, thinking about the call that the transportation is a good idea. I think it's a good idea, and it's going to make a lot of money.
Wow, that's a very fun idea. I do agree with that.
Yeah. Okay, let's go down. Is it possible to cause secondary cancer after BMT? Do you think?
Yeah, of course, that's very well established.
Okay. Will this presentation be available later? One thing I want to say, I didn't know our Zoom size is limited to 100. We bought the 500 one. So there's a miscommunication between Zoom and us. So I'm going to talk to them. That's one side. The other side, I did record this thing. So I feel very sorry people cannot come in because of the maximum size. I will make this available. I will put it in that group if you scan at the end. Dr. Pan, are you okay with it?
Yeah, sure. You can put it somewhere on YouTube or something.
Yeah, thank you. And what's the best protein source you recommended? What is the best protein?
What's the best proteins? That depends on what kind of food you like. I don't think I will be able to say like what I eat is probably very different than what everybody else eats. But the protein source, I always tell people to eat more fish, less red meat, good amount of bean. That's plant-based protein, right? Another thing is that you don't want to eat too much protein. Actually, too much protein may not be beneficial to health. You just want to eat, just eat right amount, just good enough. somewhere around 40% of your daily diet be protein. But some people drink protein powder, eat a lot of protein. There's some evidence that high protein content may actually be responsible. There was a study about a year ago that was fascinating and published in Cell Metabolism. And the study was trying to figure out the difference between plant-based protein and animal-based protein, because there's a lot of conversations about plant-based protein is better than meat-based protein, right? And the study wanted to find out, yes, then why is that? So what they found out was that it was really the amount of protein that made a difference. It wasn't really the amino acid composition in the plant-based protein source or the meat-based protein source that made any difference. The amino acid composition was similar, but the amount of protein made a difference. People who eat a plant-based protein source tends to eat low amount of protein. And perhaps that's beneficial. Eating too much protein may not be good. I eat too much protein every day. This is too much to eat every day. You have meat at lunch, you have meat at dinner.
Yeah, so I think keep it balanced. That's a perfect answer for almost everything related to diet. But we understand what you say. Yeah, it is. Yeah, how much you eat and what you eat combined.
Right.
And also, Dr. Pan, can you go out from the sharing screen? I have a couple of more slides that I want to share related to New Hope Cancer Care Foundation.
Let me start sharing. Okay.
Thank you. I'm going to add this one. I hope that's the one. No, that's not. Did you see my sharing screen? Right now you see New Hope, right?
Yep.
Okay, I know you have been president of New Hope Chinese Cancer Care Foundation for several years already. I really admire those doctors who pay special attention and who pay their energy and time to reach out to the community. So you already mentioned the New Hope Chinese Cancer Care Foundation. We really wanted to know, if we have 1 minute time, can you mention what kind of service you provide?
Maybe, how about, let's ask Tina. Tina, are you there? I'm not sure.
Yeah, yes, I'm here.
Tina will be the best person to do that.
Well,
could you give a brief introduction?
Well, yes, thank you for the opportunity to introduce New Hope Chinese Cancer Care Foundation. Mainly, we do the support and caring service to New Hope, to Chinese cancer patient and family, because when they diagnosed cancer, that's a very that's an impact. That's a great impact to them. So we do a lot of service, try to help them to go through the journey for during the treatment. So like I can mention few service like a drop-in session as an oncologist. That's a free consultation from an oncologist. That's we have it every other Wednesday afternoon, and the upcoming one is next Wednesday, August 4th. So we do it online right now by Zoom because during the pandemic. So a patient can get a free consultant for their treatment, for their opinion. I mean, it's not a second opinion, but it's a consultation. And we also offer a transportation service because during the treatment, patient needs to go to a hospital and the clinic a lot. And we try to reduce the load from the family. So we offer the transportation service. We also have support groups. We have the community education, and we do have a meal premise. A meal program is coming up. So please stay tuned with us. And thank you, Su-Ming, and for giving us opportunity to introduce us. You can find more information on our website. It's www.newhopecancer.org. Okay. Any question, you can call us and visit our website. Thank you, Dr. Pan. Thank you, Su-Ming.
Thank you, Tina. Tina's the office on and what's the title now? Well, the only person that I will go to when I reach to New Hope first is taking care of everything. And another program I would like to add on is the reaching out to educational program for cancer awareness. And now we have a lot of, well, when I say we, because I've been working as a volunteer for American Cancer Society Chinese unit, and from the day first when New Hope established. So I feel very proud of being a volunteer, and also I think the educational part, the program, if you go to their website, you will see all the video recording and very, very informative video recording. I feel very proud of being part of it. And it's just a New Hope is getting bigger and stronger. So what I want to tell you, to share the information with you today, is the annual conference that is in September 25th, from 2 o'clock maybe to 4 or 5, and it will be finalized. In there, we will be able to see how New Hope is providing patients or their caregivers help. And also we will invite patients, survivors, we will invite cancer care providers. So we will invite Dr. Tai, who's our previous president, and we will invite, well, there's a lot of things that's going to happen. So that's why I put the information here. If you want to know more about New Hope, I think you should contact New Hope directly, or you can go to that group that there's a barcode you can scan if you're interested. Not only the annual conference, I think the service they provide is really, they put patients' benefits in the top list. No matter what they do, they go around go about the patients' benefits. How can we serve them how can we serve them better? So this is an organization that I really, really recommend it. And I know we need a lot of volunteers. So we need to, if you are a company, or you are a small or large company, you want to get involved in this big whole program. When we reach out to community, you can contact them. We really need volunteers. So we also need sponsorships. So I think they will have sponsorship package over there. I really recommended you to go there. Okay, I won't, because I'm a president of CBA, of course I will remind everybody again, this is our annual conference. So it's just a 1 week, New Hope's annual conference, and then next week is our annual conference. I hope you can join both of them. And both of them is equally good to me, because I spend so much time with New Hope, and also I spend so much time with CBA. Okay, thank you. This is the group, the temporary group that I created. Everybody is welcome to join in for information for today, information for future talks, or information for CBA or New Hope. Thank you so much. Thank you.
Thank you, Su-Ming. Thank you, everybody. Have a great weekend.
Thank you, Dr. Pan. Really appreciate your time. And Tina, and all my CBA crew. Yes, thank you so much. A seminar won't be successful without your help. Even tiny little help is good. Thank you.
Bye-bye.
Thank you.
Bye.
Bye.
I'm going to end the meeting. Thank you.